pleasure to introduce Dr. Jonathan Kipnis, who comes to us not from Antarctica, but from Israel, but more recently from UVA. He graduated in 2004 from the Weizmann Institute of Science in Israel, where he was a Sir Charles Clore Scholar. Upon graduation, he won the Prize of Excellence for Doctoral Studies, awarded at the Feinberg Graduate School of the Weizmann Institute of Science. He's also the recipient of the Distinguished Prize for Excellence, awarded by the Israeli Parliament. His research has focused on the complex interactions between the immune system and the central nervous system, a fascinating area. He started to become fascinated with this as he was looking at meningeal immunity in the role of disease and health in the central nervous system. And that's what brought his team to start studying the immune cell trafficking in and out of this understudied compartment. When I asked Johnny about his hobbies, something he could share with the group, he said his secret life of the scientist was that his work and his hobby was science. He says his motto is love science or leave it. But he does enjoy a good red wine, a good book, and dinner with his wife and 3 children aged 14, 12, and 3. Tonight, Dr. Kipnis will speak with us about the immune dimension of brain function. He will explore how the immune system supports brain function and share a recent stunning discovery made by researchers at UVA that has overturned decades of textbook teaching, that the brain is directly connected to the immune system by vessels previously thought not to exist. Please welcome Dr. Kipnis. Thank you.
Thank you very much. It's a great pleasure to be here tonight, and it's not easy to speak after such prolific speakers, but I will try to do my best. And I'll tell you today about another dimension of the brain, which is the immune dimension or immune angle of the brain. So brain, as we all know, is probably the most, I shouldn't say the most important, but definitely the most complex organ in our body, composed of many regions, and there, as we just heard, are very complex connections between the regions. Brain is also a very active organ. It produces a lot of waste. The waste needs to be removed from the brain. And the question is, how does the waste is removed from the brain? Or how does the brain drain? And you'd be surprised. This is a fundamental question in science, and we did not really understand how the waste materials, how aggregate proteins like beta amyloid, which leads to Alzheimer's later in life. How does this being removed from the brain to keep it healthy? And some of you might have heard about our recent discovery about these new channels which have not been discovered, which have not been known about before. And I was told by our PR person that I should talk about them first. And I said, okay, I'll mention them first, but I need to go. You know, things don't just appear out of the vacuum. There's some order to it. So we obviously were very lucky, and we got a lot of press release on our findings. We were covered by the Washington Press, the Time magazine, more public media and more scientific media. NIH was writing about our finding, and we even got a Wikipedia page about the discovery, which was really fun. But again, before I tell you, for you to understand that this is really potentially important finding, I would like to take you on a roller coaster ride, a scientific roller coaster ride, and tell you how we got there and why we think it's important. So if you talk to an average neuroscientist, they will tell you that the brain is composed of neurons, and this is the most important cell in the body, which is true. The brain is composed of neurons, but about half of your brain, and I hope you won't be disappointed too much, is not neurons. Actually, it's made of cells called glia, which were termed glia because they thought to glue neurons together. And there are many types of glia. There's at least 4 now that we know, probably even 5, I would say. So these are the neurons, but they don't go by themselves. They're covered, they are wrapped by one particular type of glia called the oligodendrocytes, which we will not talk about them today, but they allow neurons to fire more efficiently and faster. This is the most complex type of glia called the astrocytes, and they control everything in the brain. I don't think any creature can live with, or survive or exist without astrocytes. And then there is another type of glia, which is our personal favorite glia. Those called microglia, or the small glia. And those are the macrophages in the brain. We have these macrophages seated throughout our body. So they're immune cells which are seated into our brains before we are born, and they stay there for the entire life. So they know your brain very, very well, and they stay where, and they probably are doing something important. So now if we take a mouse, which has all these cells, which are now here in blue, but they're expressing green fluorescent protein, we're making a window in a skull, and we're now imaging this mouse through a microscope, which can image a live animal through a window in a skull. What we will see is about 10% of the mouse brain will be actually covered with these green cells. And if we look in, because we're looking into live mouse, the cells are alive as well. And if you look at them, they're constantly moving. Now, if you look at any other cell type in the brain, including our famous neurons, they won't be moving. Only microglia, only these macrophages are moving. And why are they moving? What are they doing? We don't really understand very much. I think the research of these cells really has exploded in the last 5 years, and so we're just at the very, very beginning, and we're lucky to be there and study these cells. So we were studying them before 5 years ago, but we didn't know much about them. Now we know a lot more. So now if we zoom out and fly out of the brain, and so between the brain and the skull, there is the area which is called the meninges, and most people don't really care about it. But your brain is basically, it's within the fluid, right? So if you make a sharp turn, you don't hit with your brain, the skull. There are membranes there, and there is a fluid there. And this fluid is called cerebral spinal fluid. When you're getting a spinal tap, what physicians are taking is the fluid of the CSF, or cerebral spinal fluid. And this is here depicted in the blue. So this is the brain, this is the skull, these areas are the meninges, and so this is where the cerebrospinal fluid flows, and it's packed with immune cells. Those of you who have seen the presentation from my students and fellows in the lab, you actually saw the real staining for these cells, and there is lots of all kinds of immune cells, as many as you have in your gut, you probably have in your brain. Now, what are these cells doing there? We've been very, very interested about it. So let's step for a second back and ask ourselves, what's unique about many neurological disorders? I apologize that the first sentence I'm going to say will be a little bit disappointing to many of you. And the unique thing about it is that most of them, if not none, actually have a cure. Well, there is another good thing about uniqueness of neurological disorders. They also have, all of them have immune component, or most of them have an immune component. And so we believe that because the brain it's very hard to reach the brain, but it's very easy to reach the immune system. We can actually replace the immune system, we can take it out, fix it and put it back, we can actually take it out, throw it away, and give you a new and better one. We can take your cells while you're young and give it to you back when you are aged. We even have a patent on such a therapy, if it ever works. But the idea is that the immune system is replaceable, renewable, and reachable, and druggable, whereas the brain is not. So the question is then, how do we study the immune system, or how do we study the role of the immune system on the brain? Because if there is a connection, then we can actually intervene with the brain function through the immune system. And how do we first of all, how do we ask? Again, we're working with mice primarily. Oh, okay, so it says smart versus non-smart mouse. I apologize for the slides. So this will be our stupid mouse, and this will be a very smart mouse. But in the lab, we are doing it a little different. So with the experiment that we're doing in the lab is we have this pool of water, which is about 1 meter in diameter. It stands in the room, and it has signs on the walls. And now we have here the platform, which is submerged under the water. So, but if mouse hits the platform, it can stand on it, but it cannot see it because the water is opaque. Now you introduce the mouse into the pool, and the mouse starts swimming. Now mice can swim, but they don't like to swim, okay? Unless you give them very warm temperature and they think it's a jacuzzi, so they're really enjoying it, but otherwise they want to get out of the water. They're actually scared of water, but they swim pretty well. Now you show them the first day that there is a platform here, and so they look around and they should read the signs, right? And then as you introduce them in subsequent days, they learn their path much faster to the platform because they know where it's located, even though they don't see it, based on the extra-mace cues. So this is the learning. Now, if we take the platform out, we see how much time mouse spends in the area where the platform was located. And so this is basically the memory. Now, smart mouse will realize, okay, well, it was here, but not here anymore. Why don't I go and search elsewhere? And so next day, day 6 and day 7, we move platform across the street, so to speak. And so then mouse knows, smart mouse knows, okay, it wasn't here, so I'll just rather go and search elsewhere. The moment mouse finds the platform, it's very easy for it to understand where it's located. It's like your office moved basically across the street. Everything is the same, it's just one step and you are back there. So years ago, over 10 years ago, we've done this task on a mice which did not have a normal immune system. And what we showed is that mice without normal immune system cannot learn as well as mice with normal immune system. Okay, so this is the learning of a normal mouse. By day 4, they learn very fast, whereas the mouse without immune system do not learn. But if you take mice without functional immune system and you give them back this immune system, and then you check them after 2 weeks, they learn indistinguishably from normal mice. And again, this is the controls without immune system, and those did not have immune system, but now they received. Since then, we have done this experiment in different animal models of dementia and learning disabilities. We can even take an aged mouse and rejuvenate its immune system, and now show that aged mouse can learn almost as well as a young mouse. So this is pretty remarkable. So we've been interested on understanding which cells are doing it. Obviously, like I told you, I'll make now 10 years of research into really 2 minutes, and it's all published, so you can go and double check me for the facts. So the immune cells which are mediating this effect are located in this area between the brain and the skull, the meninges. So they're not invading the brain. Once they do it, it's a bad thing. That's a pathology, right? But they can do it from the borders of the brain, so they're doing it from here. And we know that because we can treat animals for only 5 days with a drug that will keep your immune system normal, not your, but mouse's immune system normal, but will prevent the cells to getting into these areas, into the meninges. And within 5 days, we see this, so this is normal mouse learning very well, whereas the mouse on this drug that doesn't allow immune cells to get into brain areas will be flatlined, so no learning whatsoever. So let me depict to you a picture of how we see it. So remember those blue cells, which then were green on a microscope, the immune cells of the brain, they do their job in the brain. And I depicted them here as trash collectors. And trash collection is a very, very important job, definitely, for the brain. And they're the phagocytes, and they collect the trash. Now, the immune cells from the periphery are just telling them how to do their job and what to do. Now, when these immune cells are malfunctioning or not working, or they just aged, then the microglia or the macrophages of the brain do not do their job properly. And so our job, this is our hypothesis. And so actually, this is a funny cartoon, but what we are actually trying to understand is we're trying to isolate now these cells from different disease states, sequence them, and understand what type of genes they're expressing under different conditions, different diseases, to better understand the picture of what these cells, what's happening with them when this immune regulation is failing. Now, of course, when we have too many of these immune cells, or they start invading the brain, then things also go very wrong, and then we have diseases like maybe multiple sclerosis, when actually these your own immune cells are tearing the brain apart. So it's not a good thing, obviously, as well. So what neurological disorders we could in theory target through the immune system, and I'll just give you a brief list. So the age-related dementia, we're actually looking in our lab, and things are very promising, including Alzheimer's disease. We're doing with collaborators brain tumors, obviously, because brain tumors need immune assistance. Immune system needs to be there to target the brain tumors. Different disorders with neurodegeneration. We're looking, obviously, in multiple sclerosis and other neuroinflammatory diseases like brain infections. And recently, we've been very interested in autism, because there are many works in autism and immune system, and how immune system is malfunctioning in autism spectrum disorder. So if the immune system are so important, it is very important to understand how do immune cells get in and out into those areas. And what are the ways of immune cells in and out? And in every other tissue in the brain, we have two types, in our body, we have two types of vessels. Vessels which are bringing in blood into the tissue, and with blood we get we get the oxygen, but we get also all the goodies, all the nutrients. And also that's how immune cells coming to survey the tissue, to make sure that the tissue is well surveyed. And then we have the second type of vessels, which are called the lymphatic vessels. And those vessels are removing the waste from all our organs, okay, back into the blood circulation, and then it's being cleared. Now, these vessels are also used by immune cells to go out. Okay, so we have two highways, in and out for immune cells. Goodies come in and waste goes out. Now, for the brain, we believe that these vessels did not exist. And so the question is, how does the brain then clean its fluid, its waste? And so the understanding was, well, from inside the brain, the waste is being cleared, or being removed into this blue, into the CSF. And from there, well, from there it could go two routes. One, through those granulations into the sinuses that drain all the blood from our brain. The idea was that part of the CSF could actually flow there, and the other path was described under the nose, actually, through the cribriform. There's a hole in the skull, and then through the nose into it. Now, immune cells or big proteins, big aggregates, cannot go through this area. Never things like this go through the blood. And so the idea was, well, then everything else goes through under the nose into these lymph nodes. So things did not work for us. We were looking in this area and we could not find much. We couldn't find any changes in immune activities when we see lots of effects here in these areas of the brain. And so I was very lucky to get a postdoctoral fellow who is actually here in the audience, Antoine Louvois. He came to my lab from France, where he was also working on immune-brain connection. And he decided to look at this, to scratch everything we knew and to start looking at it anew. And what he did is he developed this whole mount. He was able to peel the entire meninges from the whole mouse brain and peel the whole thing on the slide and label these. These are blood vessels. These are the major blood vessels called the sinuses. So the entire blood of the mouse brain and also our brain is being drained into those sinuses and then out through the jugular vein. So when Antoine labeled this whole mount with immune cells, what he found is that there were lots of immune cells in this area. All these colors are immune cells. What was really interesting is, next to the sinus, he found those strings of immune cells, very, very nicely organized immune cell strings, which were too well organized to be serendipitously there. So we realized that it must be in some sort of vessels, but we knew that these are not. There's no blood vessels right here. There's a major blood vessel here, but there is no blood vessels here. And so we decided to look for other vessels, but we knew that there is no other vessels in that area. Nevertheless, we decided to stain for markers of lymphatic endothelial cells. And what Antoine was able to show is that actually along the sinuses, we have these beautiful vessels, which are actually bona fide lymphatic vessels that express all the markers. I won't bore you with the details, but they are as lymphatic vessels as they are anywhere in the body. They express everything, they function, and they look really good. And they're actually vessels, they carry the immune cells, and the question, of course, was, are they functional? Can they really remove the waste from the brain? Can they drain the CSF? And so to answer this question, what we did is, this is again looking through a live mouse through this window in a skull, and we now inject green dye IV intravenously, and we inject the red dye into the CSF, and the red dye will mimic the waste that your brain produces and which is now removed into CSF. Now, if the CSF red dye will be drained through these vessels, means that they're draining CSF, they're draining the waste. And here you go. This is, like I told you, it's a live mouse, so we can see a live movie. This is the blood flow, and this is the flow in this one thin vessel. Actually, there are three of them, but you only see one side of it. And that is actually the drainage of the CSF, which goes out like in every other tissue, and is connected directly to your immune system. So your immune system now in the deep cervical lymph nodes along the neck, humans we have over, I think, 1,000 on each side, can actually monitor what's happening in your brain, and then if there is a problem that they can respond, get into the brain and fix if there is an infection, if there is an injury, or so this sort of thing. So we believe that many neurological disorders may be perceived or should be perceived as a neurolymphovascular diseases. And so we are in our lab are looking particularly on multiple sclerosis and Alzheimer's disease, and we believe that in this case these vessels are not functioning well enough by removal of immune cells, and so we have some brain inflammation, and in this case these vessels are not functioning for removal of big aggregates for reasons we don't understand yet, and so we have accumulation of the aggregates inside the brain. But of course other disorders may also imply. And again, so if this is the CSF and this is the sinus, these are our vessels right here. We're still looking at their exact position, but we know that they can drain the immune cells and the protein aggregates, and the function is extremely important, and we believe that many neurological disorders may be actually due to these vessels dysfunction. And my last slide, I would like to show you my lab. So these are the people that are doing the hard work while I'm traveling and speaking. And Antoine, who actually, this Antoine, he did all the work on lymphatic biology, and now he's being helped with another postdoctoral fellow, Yasmin Hertz, and Maria is a visitor from Norway, Yasmin is from Germany, and Tor is from France. We have Igor from Russia, and actually Jeff and Jim and Sachin are actually here. They're students in the lab, and some other people as well. Probably most important slide is the slide of collaborations, and most importantly, funding. Most of our funding, even though we have some private funding, but most of it is a public funding coming from the National Institute of Health, and so we owe all of you for what we do. Thank you very much. One question? Yes? Yes, good question. So it has been shown that the removal of the trash from within the brain into the CSF is more efficient while we sleep. So what we are looking now, actually just ordered the microscope for over $1 million, which will allow us to image not just a live mouse, but a live mouse while it's walking, or a live mouse while it's performing some learning task. And so we'll be able to see the function of these vessels, because like I told you, removal of trash from inside the brain into the CSF is an important step, but it's not sufficient. You don't want to have your water polluted. You want your water to be cleaned, right? So the CSF also needs to be cleaned. And so we hope that these vessels may be working more efficiently at night. We don't know.
And your theory is that that happens when we sleep?
Well, I didn't say that, but it may be more efficiently while we sleep. But the entire immune system is working way more efficiently as we sleep. That's why if you think of your kids, right? If you have kids, then they usually, when they sick, they usually will get fever towards nighttime. So immune system gets very active around nighttime when the brain goes to rest, and then the immune system takes over. Thank you.